Welcome to our biology lessons with Dr. Hanaa. Today we will solve with each other November 2025, Variant 1, Paper 6. A student investigated the effect of temperature on the movement of water in celery stalks. The water contained blue dye. So we know that when we see the word of effect of, so next the effect of which is independent variable or changed variable. In plant stems such as celery stalk, water moves in xylem tissue within the vascular bundle. The student used this method. Step 1. Take 2 celery stalks of celery and cut them so they are both 10 centimeter long. So this is one of the, yes, controlled variable. Prepare an iced cold water bath, C, and warm water bath, W. So we have here a source of error because we don't have, firstly, the temperature of the water bath, and by passing the time, the temperature change. So how can we improve this error? Yes, we have to use thermostatically controlled water bath. So in front of us, 2 large beakers, one of them C and the other is W, and both containing water. So one of them this ice cold water, and the second one with the warm water, and both containing blue dye. Measure the temperature of the water in water bath C and water bath W. Step number 4, let the blue dye equilibrate in the water bath for 3 minutes. After 3 minutes, place 1 celery stalk into each beaker, blue dye as shown in the figure. So we have here the celery stalk with a 10 centimeter long, placed in the water bath. So whatever in ice or in the warm one, with a blue dye inside the internally beaker. After 10 minutes, measure the final temperature of the water in water bath C. Complete the table 1.1 by calculating the change in water temperature for each water bath. So at the beginning, we have the first temperature will be 4, and then it becomes 15. And in W, it was 40, it becomes 28. So we will calculate the change in temperature. So by making subtraction. So it equals 11 and 12. But one of them by increasing and the other with decreasing. Step number 7, remove the celery stalk from the blue dye in water bath C and put it on the white tile. White tile, it looks like ceramic piece. Cut. We have also, this is a section across the celery stalk, the 5 millimeter from the end that was in a blue dye as shown in the figure. So I just cut it every 1 millimeter width each section. Examine the xylem tissue of the cut section of celery stalk to see if the blue dye is visible. Okay, so this is the part that's stained with the blue dye, which is xylem. Figure 1.4 shows the position of xylem tissue in a cut section of celery stalk. Step number 10, if the blue dye is still visible in the celery stalk, cut another 5 millimeter wide from the dyed end of the remaining part of the celery stalk. Continue cutting 5 millimeter wide section from the celery stalk until the blue dye is no longer visible in the cut section. Count the number of sections you have cut from the celery stalk that contain the blue dye and repeat from step 7 to 11 with a warm water bath. The students' results are shown in this figure. So in front of us, number of 5 millimeter wide sections cut from the celery stalk C, 4, but we know that the wide of each one of them, which is 5. and number of 5mm wide section cut from the celery stalk W, which is warm water, which is 13. Prepare a table and record the result. So including number of sections cut and the total distance moved by the blue dye in each celery stalk. So at the beginning, so we can get the independent variable and some of the controlled variable, and we have hazard and also error. Here we can know that the total distance moved by the blue dye, which is a dependent variable. How can we calculate this? So we know that in C, we have 4 multiply 5, because each section 5mm, which is equal 20. And we have the warm one with 13 multiply 5, which is equal 65. So the table that needed, so they need the temperature, but we don't have a specific value, just we have cold and warm. And the number of section of each, because he asked about it, the number of sections and the total distance that we calculated here. And don't forget the measuring unit, this 20mm, and this is 65mm. Why we use the millimeter? Because each section 5mm, so we have to use the same measuring unit. So this is the table, this is the final one, including the temperature, number of section, and the total distance by millimeter. Please don't forget to write the measuring unit in the heading of the table, not in the cell. Just wrote it once in the heading of the table. Calculate the rate of movement of blue dye in celery stalk in beaker W. Include the unit. How can we calculate the rate? So what's meant by rate? So something that moves in a certain time. They said that left for 10 minutes. And what about the total distance of W? Total distance of W, which is 65, and the time, which is 10 minutes. So the rate equals the total distance over time, and then we can write as 65 divided 10, which is equal 6.5. And what about the distance? Its measuring unit is millimeter, and the time was by minute. So the time, which is 10 minutes, and the rate, total distance over time, so equals 65 divided 10, which is equal 6.5. And don't forget, how can we write the measuring unit of the rate? We have this value by this way. So we have the rate, distance over time, so we have millimeter per minute. So we can write it by both ways, millimeter per. We can write it by this way, per minute, or we can write it per as a slash. So state the conclusion of this investigation. So when we describe by this way, so as the temperature increases, so the total distance increases. But we don't want to write it as a kind of observation. So the conclusion, we have to return back to the objective of the experiment. So when we write the conclusion, the higher the temperature, the greater the rate of water movement. So don't use the table to write the conclusion. We have to go to the objective of the experiment at the beginning to write the conclusion. If you just describe it as a distance traveled by the water, just like this, so it is not a conclusion, it's an observation, or can write it as a description. Next question, this investigation was only done once. So explain why it is better to repeat an investigation. We have 3 reasons. I know that there is 1 mark, but you have to know more than 1, because sometimes the question with the 2 marks or 3 marks. So why we needed to repeat the experiment? So firstly, we want to get a reliable result and spot an anomalous result, and also calculate the average. In the exam, you just write once. One of them enough, but you have to know why we need to repeat any investigation. Next question, identify two variables that were kept constant in investigation. So we know that this 5 millimeter, this is the width of each section, and the volume of the dye, length of celery stalk, which is 10, and the type of celery stalk, the concentration of the dye, all of them needed to be kept constant. So just to choose two of them while solving the exam. But when you study, so you can write all the possible options to just work your mind about it. So maybe sometimes they need more than two. So in front of you, many options. The data you have recorded in the table 1.1 may indicate that there is a source of error with the method used in this investigation. Why? Yes, what are the sources of error that in this table? So we never know that the value of temperature, and the temperature may change. So how can we improve it that we said that we have to use thermostatically controlled water bath? So at the beginning, so we have to know the exact temperature and controlled or maintained by thermostatically controlled water bath. In step 9, it is difficult to determine precisely the final position of the blue dye in the xylem. Suggest an improvement to the method that would reduce this source of error. Yes, we have a source of error here, which is maybe this 5 millimeter, it is too large to be cut as a section. Maybe the dye stop in between this 5 millimeter section, not exceeding this value, not completely cover this section. So what we're going to do, so we have to cut thinner section, or in front of us other option, we can cut celery stalk vertically and just measure the distance traveled by the dye using ruler. So in front of us, two options here. So in front of us to cut thinner sections, so instead of 5 millimeter, maybe 2 millimeter or 1 millimeter. So it depends, so just thinner section than 5 millimeter. The second option here, so we can cut celery stalk vertically and just measure the distance directly with a ruler because all the xylem vessels stained with the dye. It just measure all the stained distance. Next question, xylem forms cylinder-like structure in celery stalk. Calculate the volume of cylinder with 100 millimeter long and has a diameter 0.06 millimeter. And they give us the rule, the volume by r squared multiply h. R is related to the radius, but they give us diameter. So we know that the diameter double radius. So we can divide it into 2 to give us the radius. So how can we get the radius from the diameter? So we will get the diameter divided 2. This is the value of the radius. And they give us the length of this cylinder, which is h. And also they give us the value of pi, which is 3.14. Give your answer to 2 decimal places, and give us the space for working. So the first step, you have to calculate the value of radius. So we have the diameter 0.06 millimeter and its radius divided 2, which is equal 0.03 millimeter. Its length is 100 millimeter, and then we have to use the rule that give it to us. The volume, which is equal pi r squared multiply h, and everything that we have, 3.14 multiply 0.03 power 2, and then multiply 100. Final result, which is 0.2826. But the question here asks about the 2 decimal places, so 0.28. Celery plants can be damaged by insect pest feeding on them. This can reduce the growth of celery plant, a black aphid and larvae of the leaf miner fly are both insect pests that damage celery plant. The photograph of the black aphid, this one, and this photograph of the leaf of miner fly. So in front of us do two insects like this. Line AB represent the body length. Measure the length of AB in figure 1.6. 1.6. So you just bring the ruler and just measure its length. By measuring its length, please, you have to put in a consideration the size of the paper exam. During the final exam, the normal one, its size is totally different from the printed copy that you'll use it now, because you can find different size. So your measurement is correct. So whatever the value that you can get it, so it's correct because it depends on the size of the paper. So we can find the difference. If you just check your answer from the mark scheme, sometimes you can find this value is different because the normal size of the exam paper is totally different from the size of the printed copy that you will solve it now. So if it's just measuring, so you can find it, so it's 6 centimeter, but we don't use this as a measuring unit of the image. So you have to convert it into millimeters. So if we measure AB, which is equals 6 centimeter, we have to convert it into millimeter, as 1 centimeter equals 10 millimeter. So 6 multiply 10, which is equal 60 millimeter. Calculate the actual body length of the black aphid using the formula and your measurement. They give us this rule, but also to be easier for me and for all of you. So we have a magic triangle. This magic triangle said, I am. I am. I indicates to the image. A indicates to the actual length. And M indicates to the magnification. And then we have to circle that the needed question to be answered. So which is actual length. So equals image divided magnification. So we have to return back to the figure. So we can find it 35 times. So this is the magnification. So 60 that you get it from the diagram. So whatever your value, so it's okay if your value is different from mine. So divided 35, which is equal 1.71. But the question here asks about this one significant figure. So we can find this is one significant figure, and we can find the 7, this is more than 5. So it becomes 2 millimeter. And don't forget, sometimes they don't give us this measuring unit, so don't forget to write millimeter for the image or the actual length. And if the question asks about the magnification, don't forget to write this time. State 3 ways that the leaf of miner fly shown in figure 1.7 differs from the black aphid that's shown in the figure. So just look to these both pictures. So we can find we have here wings, but there is no wings in the aphid. And also we have, this is large eye, but this is small. And we have longer antenna here. Okay. And we have longer legs in the first figure. So I have many differences between them, many differences. So what we're going to solve it, how can we solve it? So the leaf miner fly has wings, but the black aphid does not. Leaf miner fly has larger eyes than the black aphid. A third difference, the leaf miner fly has shorter antenna that we see. This is shorter antenna like this. So we can find it here, this longer one. So you can write this comparative sentences in an opposite way. So you can write this, the black aphid has longer antenna, has small eyes, that does not have wings. So you can start with the insect that you want, but you have to refer for both insects. And the last one, the leaf of minor fly has shorter legs than black aphid. So also you can start it with the black aphid, have longer legs. So it depends on the you're starting or at the beginning of the sentences. So you can make it in a comparative way. Question F, figure 1.8 is a photograph of celery leaf. Draw a large diagram of celery leaf shown in the figure. You have to put in consideration some details about this leaf. So I needed just large in size. So the point of large, so you will take it one more. So what are the details that needed to put it in consideration? So we can find this is clearly cleft here, okay? And also the veins comes from the same or the starting point. And also we can find the outline is not smooth, it's serrated like this. And we can find one of them that folded in front of the other. Okay. And you just, if you move around this leaf by this way, it just printed in your mind. So we can find it like this folded one. And also we have another folded one overlapped in each other. So we can find that its outline is not smooth, it's serrated. Okay, so, and then we have the middle vein. When we have this middle vein, so we have different veins comes from the same place. Okay. The middle one is thicker, so we can make it any thick vein as a double line. But all of them, they're starting from the same. No problem if you fall in a mistake and can erase it. So no problem for that, but don't stress on your hand or fingers to draw it correctly from the beginning, because there's sometimes that you need to erase it. So it is not necessary to see the background drawing. And don't forget the cleft here. This is clear one. And now let you to see my drawing. This is by this way. You have to put it in consideration, this cleft, clearly. Okay. And this part, all the veins started from the same starting point, and you have to look to all the outline of the celery leaf. Next question. Amylase is an enzyme that catalyzes the breakdown of starch to form reducing sugar. Plan an investigation to determine the effect of pH, so this is independent variable, on the activity of amylase. And we know that the starch breaks down to form reducing sugar by the effect of amylase. In front of us, two ways to get the dependent variable. Maybe the disappearance of starch by using iodine solution, or the appearance of reducing sugar by using Benedict solution. So in this kind of investigation, in front of us, two ways to measure them. The activity of enzyme. So the disappearance of starch because its concentration decreases to form reducing sugar. So we can use iodine solution in the case of starch disappearance, and we can use Benedict solution in the case of appearance of reducing sugar. But in this question, we have a condition. You need to use iodine solution in your method. And now he wants this side of experiment. He needs this part. How can we plan an investigation? Just we have to bring 3 test tubes and put in each, for example, the same volume of starch suspension, for example, 50 cubic centimeter. So when talking about this is its suspension, because the starch is insoluble in water, so it looks like a suspension of the same concentration. So this considering it as a controlled variable. Yes. So you have to check it by now and then. How many controlled variable do I write the independent or dependent to be sure that you collect all 6 marks in this type of questions. So in this point, so we have controlled variable. So add to each 2 cubic centimeter of different pH. So we have very important point here. So we want to change the pH. How can we change it? And how can we maintain it? By changing the pH, you can change one of them neutral and one of them in acid and the third one with alkaline. So we want to keep the pH, so we will use something called buffer solution. So we have to use buffer solution to maintain the pH at different values. So add to each 2 cubic centimeter of different pH buffer solution. For example, a pH 5, pH 7, and pH 9. So why I choose these values? Because 7, this is the neutral, the 5 an acidic, and 9 is an alkaline. And then we have to keep the temperature maintained or constant. So we have to use thermostatically controlled water bath to maintain the temperature. So place all the test tubes in thermostatically controlled water bath to maintain the temperature. So I'm sorry that I just repeated twice. So I just place all test tubes in thermostatically controlled water bath to maintain the temperature at 37. So I repeat this sentence again. So no problem about it. So add to each, for example, the 5 cubic centimeter of amylase solution of the same concentration. So this we have it as independent variable, okay? And this is controlled variable as the same temperature, and 5 cubic centimeter of amylase solution of the same concentration, also controlled variable. So you have to write many controlled variables. After that, we have to start the stopwatch, and every 30 seconds, take a small sample. For example, 1 cubic centimeter from this mixture and place it on place a drop of 1 cubic centimeter of iodine solution using spotting tile. So what is a spotting tile? A spotting tile, it looks like something that the people that use it in drawing, okay, this is more spaces, and we can use the dropper, but calculating its volume. So if the starch is present, this iodine stays blue-black when the starch has been broken down. Iodines, sorry, iodine remains brown. After that, you have to record the time for the iodine solution to stop turning blue-black. And after that, we have to repeat 3 times for each pH and take an average. Safety precaution, we have to wear gloves, goggles, and use tongs. So when we record the time for the ideal solution to stop the turning, and we just calculate the time here, which is the dependent variable. Repetition, one of the marks, and safety precaution, one of the marks. Scientists investigated the relationship between height and the vital capacity of the lung in people. The vital capacity, the maximum volume of the air that can be expelled from the lung after taking the deepest possible breath. We have this table. How can we analyze this table? We know that the first column, which is independent variable, we have to put it on the x-axis. And the dependent variable, we have to put it on the y-axis. As it is with its measuring unit. So don't forget to write the measuring unit on the axis to take the first mark. So the first mark for the axis, label. This is the first mark. If you just write it as it is. So you just take 1 mark out of 4. And then we have to know this, the least value and the highest one. The least value and the highest one. And then you have to count how many big boxes that found in the graph paper to distribute all of these value in equally distance, then start plotting. So the second, so we have to make correct scale on the graph paper. The third one, we have to plot. And the last point and last mark for drawing the line. So with each other, this is the drawing. Okay. Height of person by centimeter should be written on the x-axis, and the vital capacity by decimeter cube on the y-axis. So I divide this, the value. It is not necessary to start with 0. So you can start it from 120, 140, 160, 180, and 200. This is for the x-axis. So it is not necessary to start from 0. So it's okay to start it within the scale that you want. And the y-axis started from 0 till 8. So it depends on the scale of each one. So maybe the independent and dependent variable not started with the same value. And then start to plot all the points, okay? And you have to put in consideration, if you just make the scale in a correct way, it is easy to plot the point in a correct way. After that, we have to join each point from dot to dot using pencil and ruler. And please concentrate about a question that some of the students asked me about, can we join the first point and reaches to the origin? يعني ask me a question to draw this part. Let's just change the color to be clear, okay? Ask me to draw this part to reaches to the origin. Is that right? No. So no extrapolation. Don't add more points. You don't know anything about them. So no extrapolation before or after these points. And then a joint from dot to dot using pencil and ruler to make this line. Describe the relation between, in your graph, between the height and vital capacity. So we can write it as the height of person increases. So what happens to the vital capacity? So it will increase. It just describe the graph. Use your graph to estimate the vital capacity of a person with a height 175 centimeter. This is 175. Indicate on your graph how you obtained your estimate. So you can get it, move up like this. So don't draw these arrows, I just to show you how can we get it. Just the intersection with your line, so we can move horizontally to get the value which is equal 4.6. So the value from your drawing. You have to show how can we get this value. In people with a certain lung disease, the enzyme elastase is more active. Elastase is a protein. State the name of reagent that's used to test for protein. Yes, we know that, which is violet reagent. Now we finished this exam. I hope that you enjoy and wait for more and more exams. Thank you and goodbye. Don't forget to like, share, and subscribe.